Sehr geehrte Damen und Herren, heute gibt es wieder mit der Übersetzung. How many got that? So, translation, part 2. So, here we go. Of course, we have to have our questions on part 1. How many amino acids are encoded by one and only one codon in the standard genetic code? Believe.
We'll just change like the fundamentals of the genetic code.
Oh yeah, why not?
Today.
Just for today.
And a lot of it's up for interpretation. You can change it in a couple years anyway.
Oh yeah.
15, 10, so what are the two amino acids that are encoded for by one and only one codon? Methionine and tryptophan. Exactly. Any other ones? No. In non-standard genetic codes, tryptophan's sometimes encoded by two codons, but methionine is only one. Why? Mutter, mutter, mutter. It's because where you start. Exactly. So that's the reason for that. How many binding sites are there for tRNAs in the ribosome? Oh. No, that's not one of the answers. What was he thinking?
It then just goes into the macro.
8, 7, 5. Okay, so what are the three sites? A, P, and E. What do they stand for? Exit, peptidyl transfer, and aminoacyl. Exactly. So, see, everyone's going to do great on the midterm. If you could just share with your friends. Yes?
So can we talk about a little bit why the exit site is considered somewhere where it binds as opposed to leaves?
So the question is, can we talk about why the exit site is a place where the tRNAs bind? And the answer is yes, we'll be talking about that for the next 20 minutes. So We will talk about translational initiation, because this is where it starts. We've got our one codon for AUG. Turns out it's quite different in bacteria and eukarya. Why would that be? Okay, so polycistronic versus monocistronic RNAs is one reason why. What's another reason why? Heard people muttering about spliceosomes down here in the front. Well, bacteria have multiple start codons, but that's an interesting aside, yes. But what happens with all of our, or almost all of our eukaryotic messenger RNAs? They get spliced. What else happens to them? They get caps put on them, and it's particularly that cap structure which is really important. Also, they get tails as well. So the messenger RNA is very different between bacteria and eukaryotes. So it's not surprising that that translation that takes place is also going to be really quite different, at least in terms of how it starts. Once it starts, the ribosomes are extremely similar, and so the processes there are really quite similar. Elongation is practically identical. Unfortunately, different names for the different factors involved, and factors are the proteins that are involved in this process, lots of Fs. And then termination turns out it's pretty similar as well. So elongation, this is basically just a review of the video that we looked at the end of last lecture. As the ribosome is elongating along the messenger RNA, it's adding these amino acids to the growing polypeptide chain. You know, here we've got that E. Now, why do you say that it's binding in the E site? We'll get back to that in just a second. You've got the extending polypeptide chain attached to the tRNA, which is in the P site. The new tRNA, the amino acid which is going to get added, gets put into the A site. You have transpeptidation that takes place. And again, this is always the growing peptide chain attached to where on that tRNA? What part of the tRNA? Maybe what end of the tRNA? The 3' OH, exactly, because that's where you're hooking up the amino acids. So the amino acid's hooked up there, the growing polypeptide chain is hooked up there. Once you've done this first step, now we have a problem, because this is all in the wrong place. And so that's where we get to binding in E sites, etc. There's a translocation that takes place. Basically, the ribosome moves relative to the tRNAs and the messenger RNA. And so now this guy is in the E site. Then we have movement of the small subunit of the ribosome, where now we've gotten back to our position up here, ready for the binding in the A site. And so it is specific binding that happens in the E site, getting back to your question here, because when you have this translocation step, it binds into that position. So the tRNA, here labeled as number 3 in purple, which was in the P site, is now binding to the E site. Okay, so how do you get here in the first place? It's great, and hopefully, again, last time you remember from where we had the video, those tRNAs are really smack dab in the middle of this complex. The large subunit and the small subunit really come together kind of like clamshells, and the pearl in the middle of it is where you've got your tRNAs and the extending polypeptide chain. So you've got to get everything together in the right place. And this is true for bacteria and for eukarya. You've got to get your messenger RNA, your initiator tRNA, all into this final complex down here. I mentioned the initiator tRNA. Initiator tRNA has methionine that's attached to it. It has an AUG anticodon. And so, of course, it's now not AUG, it's CAU, because it can be base pairing with that AUG. But it's not the same tRNA that you use for methionine inside a protein. It's actually a separate tRNA. And that's because it's got to interact with different proteins, different places in the ribosome complex. Because, as we just looked at last time, new tRNAs with the new amino acid come into the A site. This guy has to get into the P site. And so we'll look at how that happens. Again, it happens differently between bacteria and eukarya, basically because, again, this messenger RNA, the green thing down here at the bottom, is different. So you have to have messenger RNA, initiator tRNA. This all binds to the small subunit, and then only after you have this association, then the large subunit can bind. And if you go back and look at the video, you'll notice that the first association is actually with the small subunit, and the large subunit comes in very soon thereafter. So how do you get this association to take place? This is where we now diverge between bacteria and eukarya. In bacteria, there's something called the Shine-Dalgarno sequence. And it's not important what that sequence is, it's just important what the name of it is. And the reason for that is it actually varies from bacteria to bacteria. So E. coli is a little bit different than Bacillus subtilis, which is different from Clostridium, et cetera, et cetera. But that particular sequence is always present in your messenger RNA, and it base pairs with a very specific sequence in the 16S ribosomal RNA. Now, what's that ribosomal RNA? The ribosomal RNA, which is part of the small subunit of the ribosome. So the small subunit of the ribosome has a specific RNA sequence which is going to base pair with the messenger RNA at the appropriate place, usually about 10 nucleotides upstream or to the 5' end of your AUG. And because this is the interaction which is taking place, now hopefully it's a little more obvious why you can have a lot more flexibility in terms of start codons here. Because it's going to be a specific distance away from where that Shine-Dalgarno sequence is. So the actual sequence here of the codon is not going to be quite as critical. And you remember that initiator tRNA is different than the methionine tRNA you're going to be using for extension purposes. And what that tells you is that that codon-anticodon interaction in bacterial initiator tRNAs is not as absolutely specific as you have potentially with also the methionine tRNA as you're undergoing extension. So you have binding of the small subunit of the ribosome, this big purple thing, to your messenger RNA at these Shine-Dalgarno sequences. Another comment that we had was that polycistronic messenger RNAs. So you've got multiple different proteins that are being encoded for in one particular RNA, and these have multiple either ribosome binding sites. People call them Shine-Dalgarno sequences, just a different way of saying the same thing. Many times people will call these RBSs, ribosome binding sites. But each of those is going to be, again, about 10 nucleotides, 5' of an AUG. What's not on here are the stop codons at the end. Then you have association of another ribosome here, another ribosome here, etc. So this is how you end up with multiple different proteins, each of them preceded by one of these ribosome binding sites. And I mentioned this before, but partly how this works and why it's so different between bacteria and eukarya. Bacteria don't have to deal with this annoying nucleus, and so they can have translation and transcription happening at the same time in the same place. So as your messenger RNA is being made from the 5' end to the 3' end, as it's being made, that messenger RNA is going to have a Shine-Dalgarno sequence in it. The small segment of the ribosome will bind to that, and you'll start getting translation. So you could have an RNA polymerase basically on this part of the RNA here transcribing while this protein is already being made. So it's completely different from what's happening in eukaryotes, where you make your messenger RNA, you put a cap on it, you splice it, you tail it, and that then all gets exported before you have any translation that takes place whatsoever. So this is co-transcriptional translation, everything happening at the same time. It's a very efficient mechanism. It doesn't allow you as much regulation or alternative splicing. And so that's the big differences that we have between the bacteria and eukarya. So how does this all get put together? It's not just the initiator tRNA, which is here the blue thing right in the middle, associating with your messenger RNA. There also are a number of other sites which are present here and other proteins, E site, P site, and A site. Well, if you think about the small subunit of the ribosome binding to the messenger RNA, then you have to have binding of your initiator tRNA that happens to it. If you have the large subunit of the ribosome which binds to the small subunit before you have that initiator tRNA, you're going to have problems because it's right smack dab in the middle. There's no way the initiator tRNA can get in there. So there's this protein IF3, so F, factor, it's a protein. I is initiation factor. So we'll talk about initiation factors, IFs, EFs, and RFs, and TFs, but IFs are initiations, EFs are elongations, and RFs are either release factors or TFs for termination factors. So again, the molecular biologists have no imagination whatsoever. They just come up with these names, IFs. If you don't have a letter in front of the IF, that's bacterial. You have a E in front of the IF, that's going to be eukaryotic. So we'll see that in just a second. This is leading on here. So IF3, not surprisingly, is blocking the large subunit from associating until you can get that initiator tRNA bound appropriately in the T site. Turns out you don't want to have both tRNAs bound at the same time. Also in the A site, that's blocked by IF1. And again, it's making sure that the only place you can get the association of the appropriate initiator tRNA is in the P site. I skipped that a little bit already. This is now IF2. This is probably the most important. If I were doing red labeling, this would be the one that has a nice little red label on it. IF2 is a protein that first associates actually with the initiator tRNA. So again, I mentioned the initiator tRNA is different in terms of its sequence. It's different in terms of its structure because it's going to associate with different proteins than the other ones would. Okay, the tRNAs which you're using for elongation. The particular one there is IF2, which brings that initiator tRNA into the complex with a small subunit of the RNA and the messenger RNA. It is the first GTPase that we talk about in translation. There are a whole bunch of GTPases in translation. What do GTPases do? They hydrolyze GTP. Yeah, sure, okay, fine. But basically, what is that process about? So hopefully, almost Dr. Iverson talked about that already. So what happens when you have GTP hydrolysis in a G protein? Okay, I didn't quite hear that. What happens with GTPase? Okay, so it's not so much removing a base that's not supposed to be there. So GTPases bind to GTP. You have GTP hydrolysis that takes place, and that's catalyzed by the proteins bound to them. But what happens after you go from a triphosphate bound to a diphosphate bound? You have a change in the structure, and that's absolutely critical. That's what all of these GTPases are about. GTP hydrolysis has nothing to do with energetics or anything else. It's to do with a change in the structure. And it's also a timer. This GTPase happens with a certain kinetics, a certain speed that it happens with. So GTPase activity, this is a change in the structure. And as we'll see for most of these eukaryotic GTPases, it's that change in the structure which then releases the protein from the RNA. So if you have the right association, I'll get to you in just a second. So when you have the right association, so the initiator tRNA binds to the messenger RNA in the P site of the RNA. Now you have GTPase activity. The structure of IF2 changes, and that then releases from the tRNA, and now we're ready to go. Yeah?
So the IF2, I'm a little confused. Is it binding to the tRNA outside of ribosome?
Yeah.
Or like guiding it to the P site?
Okay, so the question here, again, for all of us listening at home on tape, is IF2 associated with the initiator tRNA just in this complex or also beforehand? And the answer is beforehand, and we'll look at that just on the next slide, actually. So the IF2 is associated with the initiator tRNA, and it's bound to GTP. And then when you have the association with the small subunit of the ribosome and the messenger RNA, then you have GTP hydrolysis, change in the conformation, that protein releases. And once that's released, then you can have association of the large subunit, and you're ready to elongate. On the other hand, of course, we have eukaryotes. So how do we get eukaryotic initiation to take place? As we mentioned before, always AUG. Again, there's some very minor exceptions, but nowhere near as many exceptions as you have in bacteria. Here, instead of having a sequence 10 nucleotides 5' to where you start, it's really these sequences right around the AUG. So we talked about consensus sequences before when we talked about promoters. This is the consensus sequence for translational initiation in eukaryotes. And what's important here is it's not just AUG, which you find 99.9% of the time here, but you also have nucleotides, a couple of nucleotides, either 5' or 3', which are important for this. So it's not, again, not like you have in bacteria where about 10 nucleotides away you have a ribosome binding site. Here, it's really all about that particular AUG sequence. And we'll see how that happens here in just a second. Same problem. You've got to get the small subunit together with the initiator tRNA to the start site on the messenger RNA before you get assembly of all of the rest of the complex. Here, we've got nice little E's in front of everything else. So now you know they're eukaryotic initiation factors. Unfortunately, there are a whole bunch more of them in eukaryotes than there are in bacteria. So we've got all these A's and B's, and we've got 5 as well as 1 and 3. 1 and 3 are easy because they do exactly the same thing as IF1 and IF3. So eukaryotic IF1, it's just 1A or 1 alpha. 1 beta is not important. But IF1 is blocking the A site. IF3 is blocking the association of the large subunit of the ribosome. But here, very differently from the fact that you have in bacteria, small subunit associates with the messenger RNA, now you have EIF2 with GTP bound to your initiator tRNA that associates with a small subunit of the ribosome before it sees the messenger RNA at all. So now you've got to get this complex, the initiator tRNA, the small subunit of the ribosome, to your AUG. Well, how do you do that? We've got caps, right? So first, this complex associates with the cap. How does it associate with the cap? Well, now we've got these other eukaryotic initiation factors that we don't have in bacteria. These are EIF4F, which is ridiculously confusing because it's actually made up of EIF4E and EIF4G. So EIF4F is a complex of proteins including 4E and 4G. And then we also have 4B. But most important, again, I've highlighted these in red because they're the important ones as far as we're concerned. EF4E binds to the cap. So when you have export of your capped, spliced, tailed messenger RNA, it's got a cap on it. EIF4E binds to that cap. EIF4G makes a link between the cap and the poly A tail. And we'll see why this is maybe towards the end of today, maybe on Monday. But it's very important that this be a looped structure that you have here. These additional initiation factors, which they put in here, again, this is from our textbook. This includes, EIF1A, EIF3, etc. This is now all associated at the cap. Now, the cap is not where the AUG is. In many cases in eukaryotic messenger RNAs, this can be hundreds and in a few cases even thousands of nucleotides away from where your AUG is. So what happens is you have ATP hydrolysis, which through some of the initiation factors that we're not going to talk about, causes the small subunit of the ribosome together with the initiator tRNA to what's called scan along your messenger RNA until it finds not just the AUG, but also those other sequences which are right next to it. Once that gets to the right place, you have the AUG codon, which is now base pairing with your methionine initiator tRNA. Now, EIF2, GTPase, you have GTP hydrolysis. That changes the structure of EIF2. This now dissociates. Our EIF1, which is always sitting here in the A site, our EIF3, which is sitting here in the E site, dissociate. Now you have the large subunit of the ribosome, which gets together with the small subunit and the initiator tRNA. Now we're ready to do elongation. Yes?
Why isn't all of the stuff between the cap and the start site considered an intron and spliced out?
So the question is, why don't you just get rid of that extra RNA which is there? Why is it not considered as an intron and get spliced out? When we talk about regulation, the big R after the midterm, we'll talk about that. It's also very often what's called a 5' UTR, so an untranslated region. And almost all eukaryotic messenger RNAs have these sequences. And it turns out that they're, again, very important for regulation. They're probably also important for getting other factors associated with this particular piece of RNA. And so it does seem that, again, almost all eukaryotic messenger RNAs don't have the AUG right next to the cap. They've got this extra set of sequence. And again, most of that has to do with regulation that we'll talk about much more later on. It's those small RNAs, kind of a bit of a giveaway. That's what happens a lot there. Yeah?
So the EIF and IF1, 2, and 3, same function.
So EIF, so the question is that do the initiation factors basically have the same function? And the answer is for IF1, IF2, and IF3, yes. We've got IF4 and 4F, 4E, 4G, which are clearly different. So think about tables and how you would be thinking about trying to follow these. But again, once we have the large subunit of the ribosome, which is associated, now we're good to go. You've got A site, aminoacyl, tRNA binds, and you move forward through this whole elongation step until you get to the final end of this process. We already talked about translocation. So this, not just in theory, actually in practice works. But it doesn't work very well. And it turns out that you get much better translation if you have a few more extra proteins that are involved. And it turns out that they act in a very similar way to what we already talked about for IF2 or EIF2 in terms of GTP hydrolysis. They're bound to the tRNA. That tRNA comes into the complex. If it's the correct interaction, you have GTP hydrolysis and release. If it's not the correct interaction, then you don't have GTP hydrolysis, and that particular tRNA doesn't get included. So these are now the elongation factors, EFs. So EF, TU, for all kinds of historical reasons, is the bacterial version. The eukaryotic version is much more reasonable. It's just elongation factor 1. Of course, it's the eukaryotic elongation factor 1. But it basically serves in exactly the same way as you have initiation factor 2, or EIF2, associating with a tRNA, bringing it into the ribosome. If it's an appropriate codon-anticodon interaction, then you have GTP hydrolysis and release. If it's not the appropriate interaction, you don't have the hydrolysis, you don't have the conformational change, you don't release the tRNA. And so what happens here, unfortunately I didn't show that in the video very well, but basically as the ribosome is extending, it's basically screening all of the other tRNAs which are around to make sure you get the right one that gets incorporated at the A site. And it's really pretty amazing that it's got to sort of go through all of these different tRNAs, and usually 20 plus of them, again, depending on what that codon is, in order to get the appropriate one with the appropriate anticodon. And that's probably why translation is relatively slow. If you think about all of the polymerization mechanisms we talked about in the cell so far, replication is pretty fast. Transcription is a little bit slower, but still pretty darn fast. Translation is slow. But again, it's mostly because you've got to screen through all those tRNAs in order to get the right one. Think about replication. You just got 4 bases. Transcription, the same way, just 4 bases. And so here we've got to go through 20. So it's a much, much slower process. That's the first step. Sorry, I need to move through here. The second one is getting the translocation process to take place. So remember, once you've had the transpeptidation, which takes place between the P site tRNA and the A site tRNA, now you've got your polypeptide chain attached to the A site tRNA, which is great, except that if you want to keep extending, you've got to move everything over to the P site. How does that happen? You move first the large subunit and then the small subunit of the ribosome. The way you do that is through another GTPase, also an elongation factor, conveniently elongation factor 2 in eukaryotes, EFG in bacteria. But basically, I like to think of this as the protein which basically kicks the ribosome along. So it binds in a GTP-bound form and basically pushes the small subunit out of the way, and then has GTP hydrolysis, which then pushes the large subunit along. And so binding, hydrolysis, binding, hydrolysis. And this really pushes the ribosome along the messenger RNA or pulls the messenger RNA through the ribosome. It just depends on how you look at it. But both of these elongation factors, the eukaryotic elongation factor 1, eukaryotic elongation factor 2, or EFTU, EFG, are really what gets translation working. If you didn't, again, you can get translation to work in the absence of these factors, but it's pretty wimpy. Whereas in the presence of the elongation factors, it works, and it really works quite well. One thing that you'll hopefully noticing here is that we're having lots of nucleotide triphosphate hydrolysis. We had all of our charging of the tRNAs that we talked about last time with the amino-acyl tRNA synthetases. We also now have all of these GTPases which are hydrolyzing, not just to get the right tRNA in, but also help translocate the ribosome. And we'll look at this in just a second. It turns out that all of this hydrolysis, just for getting one peptide bond, is really inefficient. So it turns out translation burns probably some of the most energy in the cell, certainly of any of our polymerization processes that we've talked about so far. So we've got initiation, we've got elongation. Now what happens when we get to the end? Now what happens is you get to a stop codon. Well, do any tRNAs bind to stop codons? Not that we've talked about so far, no. There are some, we'll talk about those in just a couple of minutes. But usually you're not going to have any association of a tRNA with a stop codon. So what happens is basically the ribosome gets stuck because there's no appropriate tRNA that binds there. Now you have association of the release factors, and factors again means that it's a protein, not a tRNA, that now associates with the A site. Then you have this termination process, and basically the termination process is fooling the ribosome into trying to make a peptide bond to water. And so now it makes a peptide bond to water. You're not associated with a tRNA anymore, because again, normally this would be associated with a tRNA which is present in the A site. Now it's not attached anymore. Your protein floats off, hopefully folds up into the right structure, and has its activity. But now we've got this ribosome with all of these extra things that are bound to it. It's got all the tRNAs that are bound to it. It's got these release factors that are bound to it. Now we've got to dissociate all of these things so that the ribosome can find the next place to translate. In the case of bacteria, it's usually going to be in a ribosome binding site, often quite close to that stop codon. In a eukaryote, it's going to be getting back to the 5' end where you've got your cap, et cetera, structure in order to get to the right place. So we also have release factors, so it's confusing. These are the RFs. So we've got IFs, EFs, and RFs. This is an image from the Watson textbook. Again, I think, unfortunately, our textbook does not do a very good job with translation. That's why I'm stealing a lot of these images from the Watson textbook. So here's what just a small subunit of the ribosome looks like. But this should be a large subunit which is on top of it. It's just been taken away to make it a little bit easier to look at. This is right after you've had termination that's taken place. Here's release factor 1 bound right in the middle next to the E site tRNA and the P site tRNA, which is here. It's just lost its polypeptide chain right here. You'll notice these sequences, GGQ and SPF. These are just amino acid sequences. It's not important that you know exactly what they are. But Q stands for glutamine. Glutamine has a nice side chain at the end, but no OH. And so you're not going to be attaching it. Whereas the rest of the structure here, again, it doesn't look like it that much here, does look a lot like your tRNA, which is there. So again, we've got our ribosome. It's all finished. The peptide's been released. How do we get everything off of it? Now, it turns out that this process is actually quite similar to elongation, only now we've got this protein stuck in the middle of the ribosome as opposed to the tRNAs, which are in there. So, just mentioned this already. Our initial release factor basically tricks the ribosome into making hydrolysis, i.e. making a peptide bond to water there. Everything gets released. We've got our polypeptide again that's going to go off and do its thing. Then we have our second release factor. This is release factor 3, again, bound to GTP. And this is actually a typo here. This should be GTP, which is bound here rather than GDP. That associates with the release factor here, which looks like a tRNA. Have hydrolysis. That hydrolysis leads to the release of this protein now, which is sitting in the A site of your ribosome. And then we have basically exchange of these proteins. Now we have an empty A site and a full E and P site. How do you get rid of those? Well, now we have another GTP binding protein that comes in, the ribosome recycling factor, that associates with our friend elongation factor G. So, binds elongation factor G. What does elongation factor G do? Kicks the ribosome, right? So, we bring in elongation factor G even in the absence of a tRNA now in the A site. You have basically translocation of the ribosome. Translocation of the ribosome, all of the tRNAs fall off. Once the tRNAs have fallen off, now you have the large subunit and small subunit that fall apart. You have IF3, which comes into the complex and blocks reassociation from taking place. So back up a little bit, talk about our termination. And again, it does a really crummy job of this in our textbook, so I'm spending a little bit extra time on it here. So we have a release factor, which comes in, binds to the ribosome where you have a stop codon. Not surprisingly, you have different release factors for different stop codons. They're not all going to be exactly the same because we have different stop codons. I'm not going to talk about all the details there, but there are different release factors that do this first step. After you have this binding, again, it convinces the ribosome to hydrolyze this peptide bond. The protein is released. Now we have GTP hydrolysis, which removes this protein from the A site. That then brings in EFG. Translocation happens normally, but normally without a tRNA in the A site. And then the two pieces fall apart, large subunit, small subunit, and IF3 is associated there. So questions on termination. How does this, and it's all about the GTP hydrolysis and conformational changes and moving things along. Yeah? Mm-hmm.
It sounded like you were saying that that binds and then DCAs happen and then it falls off, but it looks like it's staying on until the EFG moves it all the way across to the E site.
It's not so much moving it across to the E site. Basically, this is, again, it's mimicking the tRNA, which would otherwise be there. But instead of actually being associated with the messenger RNA, it's not. So as soon as you have translocation, that's going to fall off. And basically what it's doing is it's helping EFG to come in, and then you have the hydrolysis, which causes the conformational change that moves things along. Yeah?
With translocation, the EFG binding pushes along the large subunit, and then hydrolysis moves the small subunit?
It depends on whether you're, so the question is, what part of EFG is moving which parts of the subunits. We always draw it here as if binding sort of moves the small and then hydrolysis moves the large. That's an oversimplification. It's basically the binding and hydrolysis moves the whole complex along. And it's probably much more pushing the RNA as opposed to moving the subunits, because again, it's a much smaller piece to be moving there. Yeah, we always do it as in those two steps, but it's really a combination of both. Okay, so this is what those release factors, at least one of the release factors, looks like. It's probably a little bit hard to see here, but it's clearly protein and not RNA, but has a very similar structure. This end here, which is normally, again, where you'd have your OH, 3' end, where your amino acid is attached. That's what you have here on the tRNA. This has your glutamine at the end. Can no longer be, now it's not associated with an amino acid, so you get hydrolysis, basically, again, binding to water. Also has this loop down here at the other end of the protein, which is similar to the anticodon loop. So these are the structures of those, at least one of the release factors. And again, it's very similar to tRNA. Would make sense. So mentioned already that EAF4G is this sort of connector between EAF4E and the poly A binding protein. And what it does is it makes messenger RNAs in eukaryotes into loops. And this is, in fact, a micrograph down here in the corner where you can see one of these looped structures that forms when you have not just one ribosome, but multiple ribosomes that are translating one RNA. They get to the stop codon at the end here, and you have all the release factors, etc. These guys dissociate, but now they don't have very far to go to get to the cap, where you're going to have association with the small subunit with initiator tRNA that then scans along to the AUG, and you can keep going around and around and around. And so this seems to be a way that you can amplify the product of one of your messenger RNAs. So when we talked about transcription, we talked about being able to make many copies of a messenger RNA from one particular part of your genome. So that's one step of amplification. The second step of amplification is this one, where you can have multiple ribosomes bound to one messenger RNA and this whole process. This binding of the 5' and 3' ends together also seems to help for stability purposes in terms of it not getting degraded by exonucleases. And this I already mentioned before, EF4G interacts with the poly binding protein in order to get these two ends of the mRNA together. You always, pretty much all the time when you talk about translation and these things, you always show the nice linear RNA, 5' end and a 3' end. But again, they're almost always associated, at least in eukaryotic systems, together. Bacteria, that's not the case. Why? Because transcription and translation are coupled, exactly. So as you're transcribing, you're making that messenger RNA, and it seems that stability is not as critical there. Again, you don't have to export it, et cetera. Just as a reminder here, lots of NTPs are hydrolyzed per every amino acid that gets added to a growing chain. So this burns very large amounts of nucleotide triphosphates, and this is just an elongation. So charging a tRNA, you need 2 ATPs. Elongation, you need 2 GTPs, and that's above and beyond initiation and termination, where you need all of this nucleotide triphosphate hydrolysis. So it ends up using a lot of energy. Partly because of that, it seems to be that there are lots of ways that the cell controls to make sure that you don't translate bad RNAs. So how do you know how an RNA is bad? What's wrong with it? Well, one way, as we just saw, the 3' and the 5' ends are always associated with each other in eukaryotes, and that does seem to be important to get translational initiation to start. So if you had a problem with your messenger RNA, somehow it's been broken, then you're not going to have the 5' and 3' ends that are associated with each other. Or if it's broken in the middle, the ribosomes get to halfway, they're not going to get to the end. So you're not going to be making lots and lots of protein, again, at 4 NTPs per amino acid added at each time. In bacteria, there's a slightly different way that we'll take a look at this, and this is what I like to call the eat me signal for a problem that you have in bacterial translation. Usually what's happening, particularly in bacteria, is your messenger RNA gets broken. And as we talked about before, RNA, because of the fact that it's got this 2' OH in its backbone, is much more susceptible to hydrolysis than DNA. And so DNA is considerably more stable than RNA. Often you'll have breaks that happen in your messenger RNA. So what happens when you have a break in your messenger RNA in bacteria is your messenger RNA binding nicely to this extending polypeptide chain here, but then you have an A site that doesn't have any messenger RNA in it because it's just gotten to the end of your messenger RNA. Again, it's been broken here. So how do bacteria deal with this? They have this really cool sort of combination between a tRNA and a messenger RNA. So it's literally an RNA which has a tRNA structure at one end and a messenger RNA structure at the other end. And basically what it does, it will bind to the A site in what's called a stalled ribosome, and now has a amino acid that's attached to it. That amino acid then gets bound through normal peptide bond interactions here to the growing polypeptide chain, but now instead of whatever this RNA used to code for over here, now normal translation is going to translate what's present here in this tmRNA coding sequence. So it's a bunch of codons. Those bunch of codons will give you a very specific tag, and again, it's what I call the eat me tag, at the end of your protein. And basically what this says is this protein is incomplete, whatever it is over here, because it's now got this tag at the end which is gotten from the tmRNA in the first place. So that says this is not a good protein, degrade this protein. And in bacteria, it's a slightly different process. We'll talk about the protein degradation in eukaryotes, again, probably on Monday. So this is how you deal with broken messenger RNAs that you have in bacteria. We'll talk about what happens in eukaryotes in just a second, but I did want to mention first one other thing that happens in the A site. This is true actually both in bacteria and in eukarya, and also in archaea in a slightly different way as well. The textbook, they mention 20 normally coded amino acids. There are actually, I think, 23 now amino acids that people have found get associated by ribosomes, but only 20 normal codons that you have in the genetic code. The particularly important one here, it's also the most common of these, is called selenocysteine. Selenocysteine is a amino acid, again, selenocysteine, which is selenium in the position that you'd otherwise have sulfur that you have in the cysteine side chain. Critical for a number of enzymes, even for humans as well. You need to have a very small amount of selenium in your diet. Fortunately, not very much, because too much of it's not a good thing. But they also can think of these as a vitamin. So you have this selenocysteine, ends up getting added through serine, but this tRNA has an anticodon which binds to a stop codon. This gets associated with ribosomes that have this particular stop codon in them, but only when you have another signal which is present there. And this particular signal is a stem loop structure. There are different ways that you have this, but this particular one is a stem loop structure. You have this stem loop structure that brings together this GTP binding protein that acts a lot like elongation factor 1, brings in the selenocystyl tRNA to the A site. Now you have selenocysteine that's added, and then usually your polypeptide chain is going to continue here. And sometimes when you're just looking at DNA sequence, you'll see, oh, there's a stop codon right in the middle of my protein. That seems a little strange. And then if you look at it more carefully, you may notice that there's in fact a selenocysteine which has got it added in the middle of your protein, or some alternative amino acid that gets put in by taking advantage of those stop codons, which now, of course, they're 3 nucleotides. So if you have a tRNA that's going to be binding to these, you can trick the ribosome into putting in the new and different amino acid. And we may talk about this right at the end of class, but it turns out this is one of the ways that people are now doing synthetic biology, is they're making lots of new codons and anticodons and tRNAs, putting in what's called unnatural amino acids into the different proteins here. And so they use this particular process, in fact, very similar to selenocysteine here, in order to add these new amino acids and get new particular functions. There's also a neat way that people are using this to make synthetic organisms completely dependent on the addition of some of these artificial amino acids so that they don't escape and run all over the place. Yeah, you had a question? How does it know to put in the selenocysteine? You have another signal there. And in this particular case, it's a stem loop structure which forms to the 3' end of where you're going to be putting it in. And we know actually for most selenocysteine incorporations exactly what that sequence is. And so if you see the stop codon there, but you think it's actually not being used as a stop codon, you can look downstream of that and you see that there's a very specific sequence that then is going to associate with this translation elongation factor, which will bring in that selenocystyl tRNA to that particular stop codon. There are 2 other ways to do this. If you're interested, email me and I'll tell you what they are. But for this class, they're not critical. So there's a couple of other things that happen in terms of stop codons and changing of translation. This is one of my favorites, of course, because it happens in viruses. Turns out it happens in non-viral RNAs as well, but it's best understood in viruses. And it turns out it's actually best understood in retroviruses like HIV. So what you have here are 2 different proteins that are made from one RNA, but they're not made from one RNA because of differential splicing, although HIV does differential splicing too, but these are made because of a frame shift that takes place. Remember we talked about reading frames last time. It depends on where you start. Now here, in the case of pretty much all retroviruses actually, there's a particular sequence in your messenger RNA that would be translated, one particular amino acid sequence, and again, the actual details here are not critical, and then it will come across a stop codon relatively soon. However, in about 10% of the cases, the ribosome gets confused because it's got this whole stretch of one particular nucleotide. It could be U's, could be other kinds of nucleotides which are here. And what happens is this ribosome, when it undergoes its translocation, that EFG or eukaryotic elongation factor 2 mediated bumping back, it doesn't bump back exactly 3 nucleotides, it just bumps back 2. In some cases it turns out it's slightly different. But what that does is it moves your open reading frame. And so instead of UUU, UUA, GGG, it's UUU, UUU, AGG, and now you start the next codon with a G. And it turns out this makes a much longer protein before you get to the stop codon. And this is absolutely critical in the case of all the retroviruses for getting this frame shift to take place. Not surprisingly, and partly why this slide is right here, if this happened all the time, it would be a real mess. So you also have another signal of some kind in the RNA which is leading the ribosome to do this. So here in this case it's shown as a stem loop structure. There's also something called a pseudoknot that we'll talk about much more in virology next term. Monday, Wednesday, Friday, 9 to 10. Big plug for that. It does overlap apparently with the recitation section for cell biology. I'm going to talk to Dr. Rosenstiel about that. You were wrong? Okay. So I was misinformed here. Okay, fine. So Monday, Wednesday, Friday, 9 to 10. Not here, I hope. It's a horrible room. So we'll talk much more about this. But basically, this is another example of how we have a secondary structure in the RNA which is leading to different kinds of translation. In the last 5 minutes, I wanted to talk about what happens to eukaryotic RNAs that go bad. There are 2 different ways that you can have problems with your eukaryotic RNAs. Usually, the problems are that you have some kind of incomplete splicing or, again, a broken RNA that takes place. So in a normal case, which is what's shown on here on the left-hand side, you have these splicing that takes place. We talked about the exon junction complexes get added to your messenger RNA as it's being spliced. Those then get exported together with the messenger RNA out into the cytoplasm. In a normal case where you have translation, this translates, the ribosome bumps off all of these exon junction complexes until it gets to a stop codon, and you have the association of all the release factors. The ribosome comes off, everything's happy, it goes back and starts again. If, however, somehow you have a stop codon that is present in your messenger RNA before it gets to the very last exon, this means this is probably a bad protein. And usually those stop codons have happened because you've had incorrect splicing. Incorrect splicing will put you in the wrong reading frame. You'll end up with a stop codon before you expect to get there. Well, what happens is the ribosome gets to this position, but there's still some exon junction complexes which are 3' to the ribosome. This is a big red flag. It says, hey, this is probably something which is really wrong. And what happens is this signals the enzymatic process to get rid of this RNA. This RNA has got a stop codon in the wrong place before you get to the last exon. There are two things that happen. The first one is called a decapping enzyme. So it goes to the 5' end, chews in at the cap. As soon as that cap is gone, now you have 5' phosphates that all of the hungry exonucleases in the cell will just love to degrade. So get rid of the cap, gets chewed in at the end. This seems to be the main kind of regulation of messenger RNA stability that have problems in eukaryotes. Of course, you could also have a case where you don't have any kind of stop codon that happens in your messenger RNA. And what happens there is actually something really similar to what happens in the case of tmRNA that you have in bacteria. And that is that the ribosome starts translating your poly-A tail. Okay, well, poly-A tail is what? It's a whole bunch of A's. What does AAA stand for? You want to read your genetic code? It says it up here on the slide. It's lysine. So you end up with lysine, 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 lysine at the end of your protein, which is, again, an eat me signal. Says, hey, this protein didn't have an appropriate stop codon. Now, it may not have an appropriate stop codon because there's a point mutation there. Probably much more likely it didn't have a stop codon because there's a frame shift. There's some other problem with the rest of your protein. So that protein gets degraded, and it turns out there are some other proteins here. The names of them are not important, but if you get to the very end of a messenger RNA, you also have dissociation of the ribosome, but then this exosome, which we talked about before, you have exosomes in the nucleus for chewing up all the introns, but you also have exosomes present in the cytoplasm. These guys are going to chew up the RNA. They chew up both from 5' to 3', but also from 3' to 5'. They basically just go from both ends of your messenger RNA and completely degrade it. So we'll stop here because I wanted to talk about antibiotics next time. Have a nice weekend. Enjoy the sun.